The overall goal of this procedure is to construct a tool to aid in tissue identification in multi-tissue paraffin blocks. This is accomplished by first combining histogel with tissue marking dye to form a brightly colored gel plug. The second step is to process and embed the colored histogel plugs following standard tissue processing and embedding techniques. Next, a biopsy punch is used to remove the small cylinders. These are Specimen Orientation Tags, or SpOT. The final step is to embed the SpOT in a multi-tissue block with the location of the SpOT and each sample carefully documented for easy reference. Ultimately, the SpOT provides a clear visual cue for specimen orientation and identification in tissue microarrays and multi-tissue blocks.
The main advantage of this technique over existing methods such as sutures or asymmetric cores is that the SpOT does not alter the tissue and allows flexibility in the arrangement of the samples for optimal analysis.
We first had the idea for this method when we received requests from investigators to combine tissue specimens from multiple animals into a single paraffin block to save money and simplify analysis.
To prepare the SpOTs, start with adding 50 milligrams of biochemical grade BSA to 1 milliliter of tissue marking dye in a 15 milliliter conical tube. Vortex the mixture for at least 1 minute until it is completely dissolved. Next, load 9 milliliters of hydroxyethyl agarose processing gel into a 15 milliliter conical tube and heat it in a microwave at 30% power. Pulse the heat in 10-second increments until the gel is completely melted. Now, combine the melted processing gel and